All right, hello everybody. I'm going to go through our thyroid lecture here quickly uh, to give you the information that you need to know about this portion of the class. And so let's go quickly through the objectives here. The goal of this lecture is to give you a foundation about the drugs that are used to treat thyroid disorders. And we will focus on two disorders, hypo and hyperthyroidism. Okay, that's simple enough. As with all of our medicinal chemistry lectures, my main goal are the drugs. So I want you to recognize the structures of the drugs, whether we're dealing with T3, T4, Protagonisturacil, or Methymazole. I'll just call it that for now. Be able to compare the activity of those drugs used in the same class. For instance, T3 and T4. Be able to compare how quickly they work, how long they stay in the system, based on the differences in the structure. And the same for Protagonisturacil and Methymazole. Which one of the two can you use to prevent conversion of T4 to T3 in the periphery, and how they both work to prevent the synthesis of thyroid hormones in general. Be able to explain the interactions of those drugs with the binding of the drugs to the target, and there will be more of that for Protagonisturacil and Methymazole. Understand the chemical structure on the target responsible for the binding interactions, and then predict how the modification, alteration in structure will impact thyroid drug activity, potency, and selectivity. Finally, we are also going to look at the ADMET profile, specifically the metabolism of some of these drugs, and see if you can use the pharmacophore plus the structure activity relationship to predict new drugs. Okay, all right, so let me give you a quick background in about the thyroid gland, and I do that because somebody might be watching this video that's not in our module and may need to understand where the thyroid gland is at and things of that nature. So this is for the sake of completeness, but you will not be quizzed on the background part of it. Okay, so the thyroid gland is a butterfly or shield-shaped gland, and it's highly vascularized. There's a lot of blood flow, and it's a flat gland. It's located in the upper portion of the trachea. Okay, upper portion of the trachea, just below the larynx. Now, the gland is responsible for the production of two hormones, and those two hormones are T3 and T4. Okay, T3 is thyroxine, and T4 is triiodothyronine. All right. Now, there are some other molecules, other hydrogenated molecules that are produced from the thyroid gland. So the main ones that we want to focus on are T3 and T4. Those drugs are going to be transported in the blood by thyroxine binding globulin, transthyretin, and albumin. So these are three transporters that are responsible for carrying thyroid hormones in the blood. Now, T4 and T3 are lipophilic, so they have the ability to pass through the cell membrane. However, in some cells, they're actually going to require active transport. T4 can be considered to be a prodrug because in order to be active, it has to be converted inside of the cell to T3, and T3 is the active drug. T3 is going to enter the nucleus, and then it's going to bind to DNA binding proteins, and these DNA binding proteins are hormones that are responsive, and they're basically hormone responsive transcription factors. So once T3 binds to the DNA binding protein, a hormone protein DNA complex is going to form, and this is going to either hinder or stimulate gene expression. And we won't go into much details about the activity of the drugs, basically how T3 stimulates or hinder gene expression. We'll focus on the structure of the drug and the structure-activity relationship. So as I mentioned earlier, there are many different molecules that contain iodine and that are synthesized by the thyroid gland. So you have monoiodotyrosine, and so you look at the structure of tyrosine, right? Structure of tyrosine, it's an amino acid. Okay, I have a hydroxyl here. And yep, and so monoiodotyrosine basically just has one iodine that's added. Okay, so you have the mono from, you have one. Then you have diiodotyrosine, so you add you're gonna add a second iodine to get to the di, and so on and so forth, where you have a triiodotyrosine. Okay, triiodo here, and then when you look at triiodotyrosine or T3 and you look at RT3, okay, the difference is which one of the phenyl rings has the two iodine. Okay, so in this particular case for reverse T3, the outer ring has both has the two iodine, and in triiodotyrosine, the inner ring has the two iodines. Okay, so it has to do with outer ring versus inner ring. Okay. All right. So let's talk a little bit about thyroglobulin. Thyroglobulin is a storehouse for T4. It's a large glycoprotein. It contains 134 tyrosine, but only a few of those tyrosines are going to be converted and be converted to T3 and T4. Okay, so thyroglobulin binds to T3 and T4 with high affinity, and thyroglobulin will be digested in the lysosomes to liberate T3 and T4 and other iodinated compounds. Now let's look at the structure here. So this is thyroglobulin. Okay, this is thyroglobulin, and as we mentioned, you have these tyrosine residues on thyroglobulin. Thyroid peroxidase that you will see down in the lecture. I believe we're going to actually abbreviated TPO down in the lecture. So thyroid peroxidase is going to take iodine and iodinate these tyrosine residues here. Okay. Now you are going to have diiodotyrosines. You are also going to have formation. You're also going to have formation of monoiodotyrosine. Okay, so hydroxyl group here is para, and then you can have just a formation of. Let me erase that. Go back to the pen. So uh, just have it I here. Okay, and so when you have after you have the iodination that takes place, then tyrine peroxidase again is going to work on the coupling of those. So if you have the coupling of um, monoiodo monoiodotyrosine to the diiodo, you're going to have T3. And if you have two molecules that are um, diiodinated, you're going to end up with T4. All right. So the same thing that I've described here is also in words. So it begins with concentration of iodine that is going to enter the thyroid gland. Then the enzyme iodoperoxidase, that's also the name of thyroid peroxidase. It's a plasma protein, and that's going to catalyze the iodination of the tyrosine residue on the thyroglobulin. Again, I mentioned you can either have monoiodination or diiodination. After that's done, then it is going to be followed by the coupling of the iodinated tyrosine residues, and finally T4 is going to be released from thyroglobulin by proteolysis. Okay. All right. So let's look at thyroid replacement agents here. In cases where the thyroid is not making enough T3 and T4, then we are going to have to use thyroid replacing agents. Now, those agents, let me change I want a smaller pen here. Now, so those agents are going to be used for the treatment of hypothyroidism. Okay. As we mentioned earlier, thyroid hormone is going to be transported in the bloodstream, then enter the cell via membrane transporters. Once inside of the cell, T4 is going to be converted to T3. T3 is going to cross into the nucleus and bind to its receptor. Now, the T3 receptor complex is going to interact with specific sequences of DNA, which has the effect of either stimulating or inhibiting specific transcription of specific genes. Now, thyroid hormone is really a hormone that affects a lot of processes. You have development, growth, metabolism. The hormone is actually going to increase basal metabolic rate, increase body heat, increase oxygen consumption, enhance carbohydrate and lipid metabolism. Okay. Now, there's been some studies on the structure-activity relationship of thyroid replacement agents. Okay, and before I go into explaining it in detail, after the studies were done, it was found that there's been nothing more active than T3 and T4 have been discovered. Okay, so when you look at the structure of structure-activity relationship, it begins with the phenyl rings must be appropriately substituted for hormone action. Okay, what do we mean by that? So this is the pharmacophore for thyroid hormones, and the X has to be an oxygen. Okay, because you need a phenoxyphenyl pharmacophore. So the X has to be an oxygen. So I'm going to write an O here. Okay, and then from there, I'm going to draw the 6-membered phenyl ring. Then let's do the aromatic bonds in here. All right, so the R1 has to be an alanine. Okay, this is the position of the R1 here, and you know the structure of the alanine, so you're going to have 1 carbon, 2 carbons. You're going to have an amine, this position, and here the carboxylic CO2H. Okay, carboxylic acid is going to be here. Okay, so the R1 is an alanine moiety, then R5 and R3 have to be iodine, okay? R5 and R3. So where is my R5? R5 is here. It's gonna have an iodine here, and then R3 is also gonna be an iodine. Let me erase this here. Okay. R5 and R3 have to be iodine. Then R3 prime must also be an iodine. So let's draw the second. Let's draw the second phenyl here. Make sure I put in the pi electrons, and we say that R5. We've dealt with R5 and R3. Now we're dealing with R3 prime, must be an iodine. So this is R3 prime. It's going to be an iodine here. At the R4 prime position, you need to have a hydroxyl group. Okay. Now, whether or not you have iodine here is going to depend, is going to determine whether you have T3 or T4. So T3 is not going to have a iodine here, but T4 is going to have a iodine at position R5 prime. R5 prime. All right. So that's in a nutshell the structure-activity relationship of thyroid replacement agents. So what is being done is basically, if a patient has hypothyroidism, you replace, since the gland is not able to produce T3 and T4, you administer T3 and T4 to the patient. Okay. All right. So that's what I would like you to focus on here. Again, I want to note again that T4 is considered the prohormone for T3, because T3 is the actual active structure. Actual active structure. All right. Now, when you have the 5' and the 3' position that are both substituted with iodine, and you have the ring in this particular structure here, the best conformation for those two rings are to be in a perpendicular arrangement to each other. So the plane of this ring, it has to be perpendicular to the plane of that ring here. Okay. So the 3', 5' diiodothyronines favor a perpendicular orientation of the planes of the aromatic rings. So this is to minimize steric interactions. Now quickly, the available drugs. You have many different kinds of drugs that are available. But basically, you have T3 and T4 available in different formulations. So, desiccated thyroid from hog or beef thyroids, and it releases T3, T4, diiodothyronine and monoiodo as well. There's too much variability in potency, and the ratio of T4 to T3 is also very variable. T4, which again we've mentioned can be considered a prodrug, has a pK of 6.7. It's fairly bioavailable, 50 to 80%, and food is actually going to decrease the bioavailability. So the duration can be anywhere from it can be several weeks, but the half-life is about anywhere from 3 to 4 days. T4 is going to be eliminated in 50% in the urine and 50% in the feces. Now T3 can be made synthetically. You can have synthetic T3. It has a rapid onset of action. So it's used when you need both a rapid onset and the cessation also has to be shorter than T4. And so with T4 you had a half-life of 3 to 4 days and duration of several weeks. With T3 you have a duration of action of several days and a half-life of 1 day. So definitely shorter duration of action and shorter half-life there, but it has a more rapid onset. Okay, so PK is 8.4. Bioavailability is more bioavailable, 95%. It's weakly protein bound. Okay, and it's eliminated 100% in the urine. Eliminated 100% in the urine. Okay. Then you have Lyotrix. It's a mixture of T3 and T4, 4 to 1. It costs more, and it's not very clear that this has any advantage over taking one or the other of the above. All right, let's focus on the metabolism of these drugs here. So when you look at these drugs, you see different points where metabolism can take place. You see the hydroxyl ring, the hydroxyl position here. You have the carboxylic acid, obviously. You have the amine, and you also have the ether bond here, or the ether, I should say, ether bond. Then the iodines on the rings. So at the hydroxyls, you have possibility of conjugation, both with glucuronidation and with sulfation. Okay, basically you're forming glucuronide adducts and sulfate adducts. At the ether, you can have bond cleavage, so this bond can cleave at the ether. For the iodine, you can have deiodination, and finally, at the amine position here and the carboxylic acid, you can have oxidative deamination. Oxidative deamination can take place to liberate. So you have oxidative deamination here. You can also have oxidative decarboxylation here. Decarboxylation. Okay, so you can have decarboxylation here. So there's many different types of reactions that could take place. 3, 4, 5, and then 6 if I want to group all the deadenation together. Okay, so that's the different types of deadenation and metabolic pathways that can take place here. All right, so let's continue here in our study of thyroid glands. The next group of drugs that we'll take a look at are the anti-thyroid drugs. Anti-thyroid drugs. Now, the clinical application is hyperthyroidism. Okay, so when you have too much thyroid. So here you have the pharmacophore of the two big classes of drugs that we have. The thyroamides, we're going to begin with those, are major drugs that are used to treat hyperthyroidism. Okay, they are potent inhibitors of thyroid peroxidase, and they prevent the iodination of tyrosine residues. All right, and they're also going to prevent the coupling reaction. So if you remember, you have all these tyrosine residues on thyroglobulin, and those have to be iodinated first, and then have to undergo a coupling reaction before they are digested to release T3 and T4. Well, these thyroamides are going to block both of those. Okay. Now, some of the thyroamides also will block the iodination of T3, of T4 to T3. So let's look a little bit deeper into that. The structure-activity relationship. So the thyroamides are derivatives of thioureas. They are called thioureolines. They can exist in tautomeric form. Basically, you have a keto form and an enol form. So you have a tautomeric relationship there that exists. There's the 5 members and the 6-membered heterocyclics. And remember, when we talk about heterocyclic, it's basically you have a cyclic molecule, but you don't have carbons at all the positions. You may have a nitrogen here, you may have a oxygen or a sulfur, but heterocyclic basically means you don't have the same, you don't have carbon only in the ring. Okay, so Um, so you have thiouracils and thioimidazoles. Thiouracils and thioimidazoles. Okay, so both thiouracils and thioimidazoles are going to inhibit TPO within the thyroid gland. What that means is going to block the iodination of tyrosine residues within the thyroid gland. Now, only the thiouracils can inhibit peripheral deiodination of T4 to T3. Okay, and the C2 thioketo and the unsubstituted N1 are essential for activity. Activity is also enhanced by C4-enol and alkyl groups at C5 and C6. All right, so let's put this in perspective. Okay, let's put this in perspective. All right. So when we talk about the thiouracils, the representative agent is purple thiouracil. Okay, purple thiouracil. It can exist as a keto or in enol. So there's a tautomeric form there that exists, and as we mentioned, purple thiouracil is going to both block. It's going to inhibit TPO in the thyroid gland. So it's going to block. It's going to block tyrosine iodination. Okay, so purple thiouracil is going to block that iodination. It's also going to block the conversion of T3 to T4. Okay, purple thiouracil is going to block the conversion of T4 to T3. Going to block the conversion of T4 to T3. The reason why it's able to do that, two reasons. First, the C2 thioketo. Okay, you have the C2 thioketo here. The C2 position here. All right, and the unsubstituted N1 position. These are the two groups that are essential for activity. Okay. Now, activity at the C4-enol. Activity is enhanced by C4-enol and an alkyl group at C5 and C6. So, when you have alkyl groups at this is C6, C5. If you were to enhance the activity, you could put an alkyl group here at the C5 position. Okay, so activity is enhanced by alkyl groups at C5 and C6 positions. I hope this makes sense. Now, purple thiouracil is readily absorbed, 50 to 80% bioavailability. The variability may be due to a large first pass effect. Purple thiouracil has a plasma half-life of 1.5 hours. And the fact that the drug is actually going to be accumulated in the thyroid gland, it really makes the half-life of very little value, because although it's a 1.5 hours half-life, the drug is going to be accumulated in the thyroid gland, so it is going to have its effect there. Purple thiouracil is 80 to 90% plasma bound, and is excreted primarily via the kidneys. Now, methymazole. Methymazole is going to be rapidly absorbed, 93% bioavailability, and the half-life is 5 to 6 hours. It does not bind to serum albumin, and excretion is primarily via the kidneys. Now, as we mentioned before, purple thiouracil is going to block the conversion of T4 to T3 in the periphery. Methymazole is not able to do that, so methymazole does not block the conversion of T4 to T3. Has no effect on this. Methymazole is going to inhibit tyrosine peroxidase. Okay, methymazole is going to inhibit TPO and prevent, let me this here, prevent iodination of tyrosine residues. All right. Let me quickly go back here, see if there's anything else I would like to cover for you. All right. So I think I need to make something clear that will help you. So Thyroglobulins, when we mentioned Thyroglobulins, purple thyro PTU, purple Thyroglobulin is the representative of the Thyroglobulins, okay, and it's present here. It's present here. This is purple Thyroglobulin. All right. When we talk about the thiometazoles, Methymazole is the representative thiometazoole. Okay. All right. The lecture contains additional information about tyrosine kinase inhibitors. I will not cover those, and you will not be responsible for those on the exam. They're included in your slides for the sake of completion. Feel free to read it. Feel free to get familiar with it. But I will not cover those for the sake of the exam. Now, just as a general note, there is an additional class of drugs that I will not quiz you on, but I want you to be aware of. So let's take a look quickly here. If you notice, part of the reason why thyroid peroxidase can actually work is the fact that you have an iodine pump here that's going to be pumping iodine into the thyroid gland. Okay, there are drugs, mainly ions, that can be used to basically hinder that process to pump iodine into the thyroid gland. Okay, those are ions that are used. They don't really have a lot of medicinal chemistry facts that are worth mentioning, beside the fact that there are ions and they will block the pump and prevent iodine from accumulating into the thyroid gland. So I will not mention those in this lecture either. All right. I hope this is helpful. Send me an email if you have a question, and I will see you in class on Wednesday.